All right, welcome everybody, and welcome to the Friday PowerTalk Informatics seminar series. I'm very happy to introduce today's speaker, Dr. Alpert Uzun. He has a multidisciplinary academic background, includes molecular biology, biochemistry. He has masters and PhDs in computational biology and bioinformatics from Northeastern University, and he did his postdoctoral training at Brown University in the Department of Pediatrics and at Women and Infants Medical Center and the Department of Computer Science, which we just talked about briefly. He's currently Associate Professor of Pathology and Laboratory Medicine with a secondary appointment in pediatrics at Brown University, and he's also serves as Director of Cancer Bioinformatics at the Cancer Center there. His research is centered around utilizing genomics and bioinformatics to unravel the architecture of preterm birth, preeclampsia, and cancer, all complex, mysterious problems. He's got expertise in data processing, analytics, interpretation of genomic data from translational research. He also leads the development of and implementation of customized bioinformatics data pipelines for analysis of next generation sequencing assays. He and his team are actively doing comparative network analysis in different cancer subtypes, and they're focusing on designing and developing innovative computational tools related to network biology and variant visualizations. The title of this talk, Navigating the Genomic Landscape, Translational Bioinformatics Tools for Novel Drug Targets. Dr. Alpert Uzun.
Thanks so much, Amy, for this great introduction, and thank you very much for having me here, even by virtual. Thanks so much. So this is my outline of my talk, but Amy already talked a lot about my lab, so I can skip this part quicker than usual. So this is, what I'm going to talk about: complex diseases, network biology, the tools that we are developing and we developed, and how we apply into the problems in network and complex diseases, and as a conclusion, what we are getting out of them. So as we already talked, I'm a computational biologist, and in our lab, we are heavily working on complex diseases. But if you ask me which disease, then I will say 95% cancer. But of course, I have a great experience in preterm birth, preeclampsia, and we also working in Parkinson's disease. But the other thing is, as a computational biology lab, we are not like alone and working in the lab. We have great collaborators, oncologists, like pathologists, wet lab scientists, and other computational biology groups and scientists. So it's a group of work when we are working. It's not only just one-sided, because we are using the clinicians' insights, because these are the front and people they're encountering with the disease, and they have huge perspective that they can help us. And we are on the foundation side from the data management and data analysis. So it's a kind of always collaborative work in my lab. And we work in, our niche is network biology. So we are applying network biology to understand what kind of like genetic architecture behind these complex diseases. And we analyze and managing next generation sequencing data. And as I mentioned, like genomics tools development and database development is one of our section as well. And we are very interested in variant data visualization and annotation. And structural biology is coming from my PhD years, and I'm still continue on working on that, because the idea is, okay, we have those variants, and when we map them on the protein structures and we model them, are we able to see changes associated with the disease? And if we do, what kind of disease associations we can see? So this is the main picture of my lab. And of course, nobody works alone. And these are great people in my lab, that from medical students to undergrads and different backgrounds. And at the end of the day, this is a collaborative work of our collaborators and with my students. And today, I will start with the complex diseases. When we are talking about the complex disease, it is like about the results of interaction of multiple genes and environmental factors. And of course, it is about the different subgroups of patients representing different phenotypes for the same disease. And these subnetworks of a disease related with the approach that we are going to be applying for example, for finding a drug target, or finding relationships between those phenotypes. And especially after the era when we have this throughput sequencing, like whole exome sequencing, targeted sequencing, RNA-seq, whole exome, whole genome, single cell sequencing, the data exploded, honestly. Like the extensive protein-protein interaction networks started to be built, and then the databases like string biogrid, as you see here, intact. And these are not only the ones, there are many of them. So we have so many opportunities to get the interactome maps. And in the interactome maps, we able to see their interactions. And those large-scale protein interaction maps show that genes involved in related phenotypes frequently interact physically at the level of proteins in model organisms, and as well as in disease, like human disease, I mean. And these PPI networks, like protein-protein interaction networks, potentially harbor great power as they reflect the functional action of genes. And here what you see, it's another interactive map. So this is a kind of representation of in bubbles. So those bubbles are showing in different colors, and they are representing the diseases. And when we map them and when we get inside them, we can see that those proteins associated with similar disease phenotypes, let's say, have a strong affinity to interact with each other, and also have a tendency to cluster in the same network zone. So these guys playing around in the same neighborhood, but for different perspectives or for different phenotypes, but they tendency to cluster in the same network zone. So this is kind of important in that sense. So complex disease can be a good example for analyzing the network biology, because everything is interaction, like in the complex diseases. So this constructing a network, you can think about nodes, edges, and in the network biology, it's an interdisciplinary field of science, and it studies biological systems as interconnected networks. But when we talk about a node, it's a representation of a biological entity. This entity can be a gene, protein, metabolite, or whatever you like in that representation. Network biology can be applied to, for example, I'm jumping up, like medical literature. Those can be the subjects, and then you can connect those subjects with edges, like here in this one. And these edges symbolizes interaction between these biological entities, including physical binding or signaling events or co-occurrences, or again, in the medical literature, their associations for the same topic or whatever you want. So very flexible idea you can apply. So this is a network that what we see. But then I'm going to go very like quick about this, because I'm going to keep talking about these guys over and over again. So degree of a node. So basically that number of connections. So like in node A, the degree of a node A is 2 because it has 2 connections, or C is it has 3 connections, and the degree of a node is for C is 3. But then there's another concept we call like hub gene, the most connected one. So here there are like 9 interactions or connections. So we can call this node as a hub, but it doesn't have to be like one node, one network. There may be several hubs in a network. And in systems biology or in network biology, when you knock down the hub, most likely you may knock down the system as well. It doesn't necessarily, but it depends on the connections, of course. And if you have several nodes and several hubs, then you need to target those hubs. So I'm going to be coming to this point for drug repurposing or drug targets in the other next slides. So there are some key concepts of network biology. For example, like systems thinking. It is important, like the way we are working in cancer, it's the system behavior is influenced by the component of interactions and feedback loops, not just one individual behavior. So it's a system thinking. And the topology is also important. So focuses on the structure and topology of biological networks and how it affects the system function. Then dynamics, it's another important key concept for our perspectives, because it's investigating the dynamic behavior of biological networks, such as response to perturbations or changes in conditions. if you are doing a timeline experiment, you can immediately like visualize those conditions by using these dynamics and changes this in perturbations. So here I'm gonna talk about one of our tool. It is called like proteinarium. So I think the best representation why we call proteinarium is planetarium. Like in the constellation of the stars, you can all make these connections. So proteinarium, take out the stars, put the proteins in. So that's how we came up with this name. So it's a multi-sample protein-protein interaction network analysis and visualization tool. So what we do is we are basically, I can give you this intro very quickly. Instead of just having a differentially expressed gene set, for example, and then identifying this one like differentially expressed list, basically we are analyzing each patient network. If you have 1,000 patients, we are analyzing 1,000 networks. So basically, if I start like this, the data is coming from anywhere you like. It may be coming from a whole genome sequencing, whole exome sequencing, single cell sequencing, RNA-seq. So it depends on you. The beauty of this tool is there is no kind of rule that, oh, it has to be only whole genome or transcriptome, whatever you like. And you can do case control studies. You can do single sample set or just single sample, just group of proteins and visualize those networks. But the niche here is for each patient we are doing this. So I'm going to go over the technical approach that very quickly, it will give you a better understanding. So let's say this is our list and each patient is listed by a group of genes. And you may ask where these genes are coming from. Let's say if this is a transcriptomic data, for each gene, you are identifying the highly expressed genes for each patient. Sorry, for each patient, you are identifying highly expressed genes. And you may say, okay, I'm interested top 100 genes, top 200. Again, this is depend on your analysis and depend on your research question. So let's say you do have this transcriptome data. You are having this highly expressed genes for each patient, and you are asking the question, okay, top 100 genes for each patient. And then you are listing this. This may be your case file, and you are preparing another file for controls, case control, or just in the examples, just single sample set. I have 1,000 patients. I just want to see the subgrouping of those networks. But let's say if you have case and control, you are preparing two different files. Each file, one patient, and I mean each patient in one file and their genes. And the next step is converting these gene symbols to protein names. So now you have these proteins. Then the way I mentioned, like the interactome, string can be used as an interactome resource because string is including all these protein interactions that you want to use. But when I'm saying all, not like all, like everything, because we are not there yet. Still, we are investigating. So whatever we do by experiments, by computational predictions, you can get in string database. So in string database, there are three types of data. One of them is you can extract, or string people, they extract the protein-protein interactions from literature. And another one is there are some protein-protein interactions computationally predicted. And the third one is it's about coming from experimental validation. And we are using the experimentally validated protein-protein interactions. And these are the high confidence protein-protein interactions. So what we do is, okay, now we have this protein-protein interaction list here. Just imagine that these are like thousands of protein-protein interactions. This is the representative image. So you are getting this protein-protein interaction from string, and you have your seed gene list. For each patient, you have your proteins, and you are mapping those proteins. The blue colors are your proteins, the one that you are having in your list. You are mapping them on this protein interaction map. And then at this level, we are using shortest path algorithm, Dijkstra's algorithm, in order to find the most connected and in the shortest path what we can get from this map by using our own proteins and imputed proteins. Imputed proteins means if you need to connect your proteins, you need this imputed protein from the system. Then at the end of the day, you have other proteins as well, but now you are interested in only the ones that you created by your algorithm, like the shortest path algorithm, in order to see what you have. So this is done for one patient. And just imagine that you are now doing this for all patients. If you have, again, 100 patients, you are doing this over and over again for each patient. At the end of the day, you are now representing each network by a patient. So each patient, you have one network. Then we are using this Jacquard's distance just to see how those networks are similar to each other. If they are similar to each other, then their values are close to 1. Then we are using UPGMA method in order to cluster those patients by their network similarities. Just imagine that this is a taxa of like organisms. No, they are patients and they are represented by their network similarities. And then the next step is you can, after this clustering, you can now visualizing the representative network for each group of patients, or if you're interested in, you can even visualize those patients individually. But then let's say now you are interested in this specific branch. What is the network behind this group of patients? This may be a group of patients for a specific phenotype. This may be a group of patients case control study, whatever the thing is. So this is your graph, layered graph PPI network. And this tool is now available under GitHub, and you are freely welcome to use. And we publish this in Genomics. Then, by the way, this tool is installing on your machine, so you don't do this on the web. But then we got a grant from Wellcome Barrow, and we now have the opportunity to visualize with even more features and different databases in Proteinarium 2. So this is now available in on our Brown servers. Proteinarium 2 is freely available. And now we have not only strict database, we have other database like Intact and Mint database. And also we have some other new features, so like interactive features. So this is the Proteinarium story, and Proteinarium 2 is now, again, actively, freely available. But then how can we apply, for example, Proteinarium 2 for known diseases and problems? Let's say for breast cancer. This is one of the applications that I'm going to go over, not in detail, but just give you the impression about how we can apply this. So this is the mismatch repair protein deficient breast cancer problem. So if I give you like intro about this, like literally every 9 seconds, a woman is diagnosed with breast cancer worldwide. And MMR is mismatch repair is vital component in cancer management, extending beyond breast cancer to include colon and endometrial cancer. And understanding the inner workings of MMR is key because it involves 7 crucial proteins, and those proteins I listed here, MSH2, MSH3. Together, they form a powerful defense against genetic mutations. And these mutations impair MMR functionality, lead to translation loss, or subsequently causing genomic instability. And surprisingly, the precise role of MMR in breast cancer remains largely or not well identified or unknown, let's say. And specifically, 2 to 3% of breast cancer patients show signs of MMR deficiency. It's not like high, but still it is a important fact. So what we did was we used data from TCGA, and we used the CBI portal data, and this is specifically for 29 MMR deficient patients and 965 MMR intact. So I'm not going to go into the details of the steps, but what we end up by 32 MMR intact and 29 MMR deficient patients. So what you see is here, this is the dendrogram, the one that I showed in the previous it was the like more rectangular one, but this is when your lack of space, it's best way to show in a kind of circular way. So the blue colors are intact, 32 intact patients, MMR patients. And then the yellow colors are 29 MMR deficient. So the network representing intact patients are here, and the yellow colors are represented by the intact patients network is here. And what you see is there are some hub proteins here. These hub proteins, especially in the MMR deficient, heavily associated with histone proteins. And then what you can see is the hub proteins are histone, and then some other proteins associated with the like the hub proteins as well. And you see here another hub proteins from the intact. So what you can use those proteins for example, they can be served as a drug target. And in our paper we published last year, we talk about this in detail. But the idea here is, without identifying any those groups, now we know those groups representing a specific network for each different entities and phenotype, I can say. So this is a kind of applicable approach for drug targets, for identifying the phenotype of the disease, and so on. And another interesting application is on glioblastoma. Honestly, we did so many applications, and I'm just showing some of them. In glioblastoma, what we did was, we used again data from available data resources, but let me go over the introduction of this. So it's an aggressive, all maybe know, aggressive brain cancer originating from glial cells, and it is the most common and malignant primary brain tumor in adults with a poor prognosis. And GBM has genetic alterations including IDH1 and IDH2 mutations, TP53, RB1 loss, and EGFR gene applications. And treatment involves surgical resection, radiation therapy, and chemotherapy. So what we did was, but before what we do, or what we did, let's say, we are still working on that project. Charissa and Jennifer, these are two brilliant students. Charissa now in Columbia Medical School, and Jennifer Lee is going to be graduating next year. So they work on this project. They did their creativity. They did very hardworking in that project, and we end up by some significant results. So we used two different populations, but three data source. One of them is cBioportal. We have 166 patients with the RNA-seq data. This is coming from Firehose Legacy in the cBioportal. So what we did, or what you see is here, there are 100. We asked the question, okay, bring me the list of the beginning of proteinarium. Bring me the list of, for each patient, top highly 150 genes. Why you ask top 150? We look for top 50, top 150, top 200, up to top 300. So there's another approach called Copernicus score, but I'm not gonna go into this, but we end up by top 150 genes representing our patients perspective in a better way. So that's why we went through for each patient top 150 genes, and we built those networks. So what you see is here, again, this is the dendrogram, and each edge of the dendrogram is one patient. So this red color cluster C15 is group of 54 patients. They came in group red color because of their network similarities. And then the blue one is C120. And here C68 is another network that we identified, and we want to go in deeper. And then when you, so here, what you do is asking the question is, and there's another approach called separation testing. We are asking the question about those networks, how they close to each other, and if they are closing to each other, what are their distance. So what we find out that C15 and C68 and C50 and C120, at the end of the day, C15 is the most distant network or cluster from the other patients. If I go back here, you will see that C15 is this group of patients. And I'm going to go into the network as well, but I just want to show you that C15 is the most distant network from the other two groups. And then what we did was, since we have the CBio portal data, we went back to this patient phenotype. What is the phenotype of this patient? So C15 is here. What you see is this is the median overall survival. It is the least survival one. And then disease-free survival is for C15 is 4.76 months. And again, from the phenotype information, the disease phenotype, so you can see how this fraction genome altered in a quite significant in C15 than the other ones. So here, what we have now is we have these groups and we have the network of these groups, like the clusters. So now we can look for the drug repurposing, where we can look for the like the hub genes in these networks. So this is C15 network, the one that you see those patients, 54 patients represented by this network. And the red colors are imputed from the system. The blue colors are it is coming from our seed genes, from our list that we put in the system for analyzing from our patients. And the bigger size means they have more interaction. So for example, TF, it has like 2 interactions, but according to FN1 is bigger because it has more interactions. Then we ask the question, are there any available drugs for any other purpose? So for those specific targets, FPR2, GPR84, and PTFR, and then the other one, for 5 of them. There are drugs, and we can use these drugs for repurposing. So the idea is now we know where those targets are. We can use these drug repurposing for at the same time or one by one, just to collapsing the system, and in a cell line, we can visualize in vivo if those drugs can be useful for that purpose. And then the other network, C68, another drug target that we can use, or drug targets. I'm not going to go all these drug targets, but you can read. There are specific drug targets for other purposes, but we can use those targets for our own purpose. That's why we are saying drug repurposing. And then C120 is another network that I mentioned. This is the network of this cluster. And then there are, in this one, there is one drug target, HSPA8. So we use this data set, but then there's another data set, Chinese Glioma Genome Atlas. And here there are 2 different data sets. One of them is with 140 samples and the other one 85 samples, and they are all RNA-seq data. So I'm going to go quickly in that part. So you see those different colors of clusters and their representative networks. And there's another data set, the second data set. They are less patients, 85 patients, and their clusters and their networks. And they are the number of patients you can see here. So the thing is, are we able to see similar network behavior, the one that we see in the first data set? So when we do this comparison, we look also for the overall survival days and potential, networks in this data set. So we find out that C20 is the lowest survival net. And then when we compare these, for example, in their own groups, this is what you see in the survival for each network. What I'm saying is for these 2 networks. in this network. Then the question was how they close to each other from our first TCGA data and here. So we find out that C15 from the first data set in the Chinese glioma data set, C20. In this network, there are 9 genes shared. And we did some computation analysis. Is it random? So whatever you took, are you always going to get 9 genes or more than 9 genes? No. In even very random analysis, what we did, we find out that you can go up to 7 or 6 genes that they can be shared. So this is not random. So then we ask the question, let's see, there's comparative analysis, their survival days. So C15 is the lowest one, and C20 was on the lowest one on the on the Chinese data set, Chinese glioma data set. So these are the representation of comparative analysis. Then what are those 9 genes? So these are the 9 genes, and these 9 genes are associated with angiogenesis, cancer progression, including metastasis. And now what we can do is, so these 2 different data sets with the similar network and 9 overlapping targets can be used for drug repurposing. So this was another outcome for that purpose. So the question is, are there drugs for those targets? Yes, there are some drugs. For example, PTFR, you can end up by 3 different drug repurposing approaches. Then you can use this in the like the wet lab experiment. So that's why we are collaborating with wet lab scientists. So this was the proteinarium story. So I'm going to be switching gears about another innovative bioinformatics tool. So we call this variant graph craft. I don't know if you know Minecraft. So in the Minecraft, you are building like worlds, like the worlds of free will. You can create whatever you like. But in this one, we are crafting the graphs. So again, a little bit network biology approaches here. So what we are dealing with now, like the VCF files, the variant call format. So I just want to talk about first, what is VCF? So genomic data holds vital insight for us, right? And then at the end of the day, for example, when you are dealing with whole genome sequencing or whole exome sequencing, the end file is the VCF variant call format, VCF file. And this is the file looks like. Quite scary though. So if you think about, like, it's a whole genome sequencing, you have all these 20,000 their variants, maybe 7 million variants or whatever the million of variants in the rows. And in the columns, if you have 1,000 patients, 200 patients, it's a huge matrix, and it involves different type of data. It has variant positions, it has alleles, genotype calls, quality scores. Basically, it's a text file, but it's a very dense file. And it is carrying valuable information. And basically, there are challenges. For example, these VCF files have extensive data richness. And when you are visualizing interpretation, data demands on different levels of associations. For example, you need to first filter, extract. You need to get this phenotype annotation information if you are interested in. Then you are plotting. As you see, there are different tools, different steps. By the way, I put this cartoon. So besides being a scientist, I'm a professional illustrator and cartoonist. So if you go to Biocomicals, you will see a bunch of biology and medicine related cartoons. So I'm reflecting our days of lives in those cartoons. So if I go back to those steps again, so you will see like different tools, different options, and different steps, and you are dealing with a huge complex data set. So we came up with this tool. We call this variant graph craft. The very first idea is we are dealing with this complex text data set, and since I know I'm heavily dealing with, you know, a pathologist, oncologist, like a molecular biologist, biologists, so they don't want to spend on time these huge complex text files. They just want to get the results. So this tool is, first of all, very user-friendly. And these two guys, Jennifer and Andy, they did a great job. Jennifer was the lead developer in this tool. So what we did was we asked the question, okay, if you have this VCF file and if you want to bring a specific gene of interest or specific genomic interest, how can you visualize in a very friendly, faster, and secure way? So this was the answer for us. So we developed this variant GraphCraft. So basically, the idea is you have a query pipeline. You can search queries of genomic locations, or you can ask multiple genes. Maybe you are interested in specific genes, group of genes. You can submit this group of genes as well. Or you are interested in multiple genomic locations. You can ask the question. So first, we are getting the VCF file. We are creating these info and indexed internal files. Then the next step is, based on your question, you are extracting the information about variants. And then you can visualize in different ways. I'm going to go and showing like hands-on way of visualizing data. So you can visualize bar graphs, not graphs. You can download the variant of interest in an Excel file. So a bunch of other important tests that you can do easily. And we are integrating different databases, MSIG database, CleanVar information, Nomad, and we specifically looking for 37 and 38. So creating this availability for the researchers that they don't stuck with only one genome assembly. And there is no cloud uploads, ensuring data privacy. So VCF is important file because you may be carrying some important specific patient information, even if it's your research, or it may be a clinical information. So you don't want to upload your data somebody's computer, basically the cloud. And then users can work with their genomic data in a secure and confidential setting, which is your own servers and your own machine. So the idea is installing this machine on your own computers. And we create packages. It's easy to install. You don't need to be a computer expert. Just click of a button, you can install this machine. Sorry, this tool. And then users can search for specific genome locations, as I mentioned. And we are supporting the pathogenic variant visualization. And also you can include a phenotype information. Let's say you have different group of patients. They may be cases or they may be different status of the disease. You can upload this phenotype information and we can visualize this too. So what about application of this? So previously we did whole exome sequencing and we did for preeclamptic and term mothers. And this was a very good example for us how to visualize this data. So in this data, we have 143 samples. 61 of them are early onset severe preeclamptic cases and 82 term mothers. And we published this, this whole exome sequencing analysis and outputs in a different paper, but we use these samples and we see a file, how we can visualize this. So total of 702 variants in this VCF file. And you can see the file after filters, 2.45 gigabyte of data. Just imagine that this is a huge data. And you are asking this question, okay, bring me the BRCA1 gene. So this is the immediate analysis. Okay, this is the specific locations. If you click on these, everything is interactive, by the way. If you click on this interaction for this specific bar, you may be immediately seeing, you are seeing immediately the variant. You will identify the variant information. If it's a benign, it is benign. If it is not, it will be same pathogenic. And then you can go even more deeper. And on the side of the screen, you will see nomad interactions. You can have more information about that. Then another question is, okay, now I have BRCA1, but out of this 2.45 gigabyte of data, I just want to go only for BRCA1 variants. You can visualize this variants and you can, by the way, it says 48, but it didn't go because of the screen capture, it didn't go all the way. There are 57 variants associated with BRCA1 gene. And then you can save this list as an Excel file or CSV or PDF or whatever you like. Or you can protect this sheet again, because this is valuable information. This is specific information about those patients and their genomic data. Or another question, I just want to visualize this BRCA1 with a graph visualization. So now every blue dot is a variant, and green and red colors are group of cases and controls. The red colors are the cases, the green colors are controls. And you can visualize this in any way, in any color you like. It doesn't have to be like in this way. And then everything is an interactive. If you click a note here, you will immediately see this is benign, and also this is more information about the specific NOMED information. Or if you click on those patients, you can see immediately patient ID there. And another thing is 3D of this visualization. So everything here in this presentation is static, but in the actual tool, everything is interactive. So you can go behind this variants, or you can visualize how those are interacted. And there are several information, but I just want to give one more thing. And what about pathogenic information? So you can visualize, okay, give me only pathogenic variants and their association with the patients. You can immediately see what are they. And then what about comparative genotype analysis? So you can, by the way, here, what you see is the all 57 variants, and the light color is the depth of variants, like read depth, and the darker color is more deeper reads, like in the ocean. If you go deeper, it is darker blue, and on the surface of the ocean, it is lighter blue. So you can immediately see those variants associated with the, genotype information. You can see what are the heterozygous, homozygous variant associations for this specific variant. Again, everything is very interactive. And then another question is, what about not the gene, but you are looking for a specific genomic location in the chromosome 17, give me the information. If there is one variant, one variant. If there are more variants, whatever the information, bring me this information. It will bring you this information. This is the variant, these are the patients, and you can see homozygous reference, heterozygous reference, or homozygous alternative variants represented here. But if you ask the question, only give me the heterozygous variant associated with those patients, you can see that one too. Or only homozygous alternative variants. So you can see those specific patients associated with this variant of interest. Then another question you can ask, give me the gene data. So for BRCA1, you see there are those pathogenic variants. There are benign variants. They are coming all from ClinVar, up-to-date ClinVar database, and no consensus ones. And also the other question is, you may be interested in specifics of those. It may be genes or gene, because you can ask several genes and you can list it in a tab here. But you can also ask the question about what are the biological processes, cellular components, like what are the pathways associated with this gene. Immediately you can get this. The idea is, from a very, very dense populated file, are you able to extract this to the target information? And then, as a summary, it's a comprehensive solution, and user-friendly interface, and custom queries, and integrated information that you can create your queries based on your needs, and the enhanced visualization tools. So I'm a computational biologist, but I'm also a molecular biologist. I did experimental work on the wet lab. So that's why I try to, in our lab, try to make very, very user-friendly in order to make the life easier, because you don't want to deal issues about like coding or downloading. You just want to go to the point. So that's why this is one of the important facts, that when we are talking about user-friendly interface. And again, this is on the GitHub. There are 3 versions: Linux version, Mac version, and Windows version. They are all freely available. There's a huge GitHub information that how you can use in detail, and you can, again, install on your machine easily. If you have any questions, or if you want to, ask about any features, you are very welcome to email me, and I will be happy to, help in any way for Proteinarium, for variant GraphCraft. We are calling VGC, by the way. And the last, I can say this as a tool and web server. And this is a very good combination of Proteinarium and cancer and how we can visualize this data. So we call this EPIC, not the APIC one in the clinical setting. So EPIC or EPIC, Atlas of Protein-Protein Interactions in Cancer. So what we did was we asked the question, okay, what if we visualize all the potential available networks for all these different cancer types? And if we create a web server and make an open source, and for the researchers, they can get in and get this information for their own perspectives. So this is the huge data, and what you see is the whole network of everything, what you see, all those patients. So sorry, for cancer types, different color, different cancer types. And then in this, like the kind of interactome of our cancer genome, let's say in this set, you can see where those networks existing. But then the question is, what if you just want to visualize them? So APIC is answering them, or answering this question. So we have very talented students. One of our talented students built all these images. And again, these images are, everything is interactive. When you click any of those, for example, images, it is bringing up, or like the drop-down menu. So let's say if you are interested in the breast cancer, when you ask the question, are there any available networks associated with breast cancer? Yes, we did for ductal, lobular, and MMR intact and MMR deficient, the one that I told you. For example, for this MMR deficient BRCA data, what you see is here, when you click, immediately, again, everything is interactive. You will see the potential network for these 52 patients. This is the representation of this network. And the red colors are drug targets that you can use for drug repurposing. And as long as you are visualizing this, everything is interactive. If you click any of the protein here, immediately you will see, for example, human protein atlas information about this specific protein that you clicked on. Or you can see ATNC information. Or gprofiler is asking the question, okay, this is my network. Annotate this network and what kind of pathways associated with this network. Then you can answer this network, and by the p-value, you can identify if it is, or how significant this network is. And then again, CREW is coming from Broad Institute. This is the drug repurposing resource. So there are a couple of different drugs here and their targets. So you can also ask the question about, okay, bring me the drug targets and drugs, repurposing drugs for those targets. Then another interesting thing is, I told you, like, this network is about for 52 patients. So these 52 patients are coming from CBioPortal data. And you can download this patient data. You can download this patient clinical data only specific to these patients. And then you can ask the question that I was showing about the glioblastoma, like, okay, what is the phenotype behind this disease, phenotype behind those patients, why those patients came together for this network? This is network, but what about their clinical data? You can visualize all this information. And this is, again, not one person job. So this is a great team, because you will see, like, how can I say, you will see residents, you will see fellows, undergrads, medical students, and my collaborator, Dr. Uzun. They work together. Everybody was really building this from different perspectives. And also, again, nobody works alone. We have great collaborators. And these people are molecular biologists, clinicians, and also pathologists. So it was a kind of very productive project from not only combining the information, but also bringing up people together. So this EPIC funded by the Legorreta Cancer Center and Rhode Island Foundation grants. And at the end of the day, basically the idea was how we can make this efficiently useful for the people who are interested in cancer, network biology, variant, and data visualization. So Proteinarium now has two different, actually not two different versions. Proteinarium 1 is the initial one. Now Proteinarium 2 is the most extended one. And we are, as I mentioned, we are identifying cluster of patients and visualizing their networks. And variant GraphCraft is recently out. It is available again. So you can use on your needs. If you have any questions, needs or suggestions, be very, very welcome. And EPIC is again available. It is, I think I put the information here in the EPIC. Or maybe I don't know. But EPIC is also, if you just say Brown EPIC, it will be coming up. And this is the team from my group, Genomic and Machine Intelligence Lab members, and my collaborators from Brown, Yale, NIH, NYU, and also these are the fundings that we got from Legorreta Cancer Center, NIH, Borough Wellcome, and Rhode Island Foundation. Again, nobody works alone, so this is a combination of all our work and it's a team effort. And thank you so much for your time. So if you have any questions, any comments, very welcome.
There are a couple of comments and questions in the chat. I don't know if you can see them. If not, I can also read them.
Yeah, let me go very quickly. Yeah. So one of these interesting, in this translation from transcript name to protein and to the latest network, is there consideration of protein modifying enzymes and their impact on the validity importance of the network? Separately, how do you include localization of the component proteins in the network? Yeah, these are the second questions. Very, very good question. That's what we are working on now. But for the first question, we didn't think about this way, but this is for the validity and importance of the network. I agree with you, like protein modifying enzymes, maybe it can be a guidance for a how can I say, like a wet lab setup, but we didn't apply that. And how do you include localization of the component proteins in the network? This is a great question. So the other question is, for example, for cancer networks. So when we annotate them, we are ending up by very, very usual terms. Okay, these are the cellular components, but whatever you do, always getting similar. But it's not cancer, or let's say if it is a bladder cancer, it's not bladder cancer specific. So now we are working on a new database. We are annotating those cancer networks and identifying their compartments, including their biological functions. So if you have a network without knowing anything, are you able to name it? Oh, okay, this is a subnetwork of a bladder cancer or of a breast cancer, and heavily compartmentalized in this specific, for example, cellular components. So that's what we are working on now. But in these current versions, only we are annotating them in generally. The second, I don't know if I answered your question, but if I don't, please let me know. And the second question, variant GraphCraft VGC, how big a data set can this handle? 100 samples. This is a very good question. So we end up by 20 gigabyte of data on your own machine, but also the other important thing is it depends on your machine's power. You can install into a server and maybe it will be a faster version. But we went up to, I think, maybe 400 patients. But the thing is, for example, when heavily dense file, let's say 20 gigabyte of data, when you first upload this file, the initial analysis is kind of small because it is indexing. But if you open this file in the next run, it is faster. Because the idea is for the VCF files, you are not only dealing once, you are opening, dealing, and okay, I'm done. You don't do this. You are keep running this again and again in a couple of days, or you are coming back to this file again. So our idea was the initial starting is slow if it's a very big file. Then the next time, since it was indexed, it is faster.
A quick comment that we are over time, so
Oh, okay.
I don't know if you have time to stick around to answer these couple questions, if
course.
and I don't want to make anybody feel like they're obligated to stay. If you need to leave for another meeting, we totally understand, but thank you if you choose to stay and hear the
Yeah.
answers to these questions.
Yeah, and if you have any questions, I have time to answer, but if you have hold on, it's totally fine too. Oh, the other question is intrigued by the Elaine protein that degrades collagen and elastase. Turns out that peptides so generated have separate function from parent protein. Been studying this for the past 20 years. Collagen?
Yeah, can you hear me?
Yes, I can hear you. Yes.
Good. So this is the Lung Health Center discovered a tripeptide that comes from collagen, and it's the basis of I think about $20 million worth of research and ongoing. So it's really intriguing when you have the protein. It's looking at what the proteins are doing that then is most relevant, because just because they're there doesn't mean that they have a functional outcome. In this case, the functional outcome was completely different from what you would have expected if you just took if you look at collagen, you say, what does collagen do? Well, it binds things together.
Right, exactly.
But it turns out the collagen peptide that was generated is actually the pro-inflammatory mediator of heart disease.
No, that's
so we have to get, obviously you're working towards this. So you've done a translation out of the transcriptome sort of environment into the protein environment, but the protein environment is a whole, there's many, many more complications, whether it be protein modifications, protein mutation, and protein degradation.
very true, very true. And the other thing,
I admire you for trying this. This is enormous, and going graphically is the only way you can start to absorb all that data, because it's a tremendous amount of data, as you well know.
exactly.
And then plan the next thing that you need to do.
Right.
So,
the other thing is, for example, we are trying to look in the very beginning of my talk. I was telling you we are looking for the close neighbors. Of course, you can extend this network and then you can see the 6-degree connection. But like in the environment of friendship, we are just looking friend of friends, not like the 3-degree, 4-degree. Because in this one, it gives us more localized focus. So definitely, I will tell also my colleagues about that, because now the next stage, especially for glioblastoma, we are now collaborating with wet lab people. So now they are going to be those targets in order to see if those drugs going to be really like effective in this silico environment.
right. Whilst you were presenting, I was building a network. So I do transcripts to metabolites. So, metabolites are an outcome and a regulator. In the old days, we used to think that, the message just flowed down from the genes and it didn't go backwards.
Right.
But we realized that the metabolites sometimes have very strong impact on what's going up much further upstream. And so I'm working with the people who are studying acute kidney injury and trying to build networks for them.
Oh, this is now remind me. So we have another collaboration. So this is about the DNA repair, enzymes, ovarian cancer, and metabolites, how we can see. So basically, similar idea, but we are now looking for the metabolites interactions in the networks.
Yeah, yeah. Well,
I
very
don't-
good. I mean, I wish I had a team like you. I've been trying at UAB to get people to move out of their places of comfort. So there's a lot of yeah, I was, by the way, 10 years or more ago, I was on the NIH Council of Councils, where we funded single cell science. So I have a very strong interest in single cell science, which we're now moving much more towards imaging mass spectrometry
Mhm.
because of the locale issue. So we need to know where proteins are, where metabolites are, and
That's good, yeah.
we had a meeting 2 days ago. It was virtual for me at NIH, and there was absolutely amazing stuff that was presented by people from Northwestern and from WashU.
Right. And the other interesting is about the single cell sequencing. What if those clusters and their specific networks? That's what we do right now. So we are asking the question, like the way you are describing, like the spatial and specifically for those clusters, in what part of the tissue are we able to see any networks? And then I don't know if you know Rob Sabol from he came to Brown last year. So he was at at your university, but in Mobile. So now I'm collaborating with him for this, like this DNA repair in ovarian cancer. So I agree with you because especially the last topic that you mentioned, now it is more important where to look and specifically what kind of, because when we're getting the tissue, for example, I did this whole exome study. We collect blood samples from preeclamptic mothers. We are just getting the blood sample. But what if, for example, if it is a placenta, if we get the placenta in different parts of the placenta, are we able to tell anything about the placenta by, for example, single sequencing? We didn't do it at that time. But I think the field is going in that direction.
Yeah, it definitely has. I was doing imaging mass spec about 15 years ago. I don't know whether you know Richard Caprioli, but he's at Vanderbilt, and he sort of pioneered imaging mass spec. So we were looking. I had a model of cataract disease in a rat. So we were looking at the lens of the rat, and we were looking for the location and abundance of proteins that are chaperones. So I don't know whether what you know about lens physiology, but you basically stop making the proteins in the lens the day you were born.
Right, right.
And they are going to be with you for the rest of your life. So around about 50 years of age is when the chaperones start to run out of their capability of keeping the proteins in solution.
Right.
And that starts the cataract process.
I think sorbitol metabolism, right? The sorbitol.
Yes, you're correct. There are different things. Glutathione is a very important one, and there's an ascorbate glutathione cycle that's going on. But you just get cumulative damage because you don't get any replacement.
Right. When I was doing my master's in medical biochemistry, I did aging in lens. So we were collecting those lenses. So in the lenses, they don't get the intact. They were just squeezing from the patient, and we were getting the lens tissue, and we were looking for the sorbitol metabolism, how it was.
Yeah, interesting. Yeah. So we then moved on to doing lipid imaging chemistry in this case in ischemia reperfusion injury in the kidney. And by that time, we managed to get down to what appears to be single cells. We can actually see single cells and see which ones have which lipids in the cell. And of course, we also means that we could see this on a grand scale. So I think that's integrating all of these things together. So we imaging proteins, imaging lipids, imaging metabolites, knowing where the transcripts are, and then you've got modification of the transcripts. got some other data that we're having to look at today about methylation of transcripts. And I don't know how well, I'm not a RNA DNA person, so I don't know how you distinguish between a methylated transcript and a non-methylated transcript. Is it done directly, or do you have to do a sulfate reduction? Or
we did this in preterm birth mothers methylation status, and we did methylation basically by sequencing. So we identify those methylated regions and we published this paper. And if you're interested, I can send this paper as well.
Yeah.
This was
So you use, is it bisulfite or
bisulfite. I think I tried to remember, it was bisulfite. Yeah.
Right.
I think it was maybe 6 years ago, probably.
Yeah. So the methylation demethylation is controlled by metabolite. So you've got acetyldenosol methionine putting methyl groups on, and you've got alpha ketoglutarate, which is a cofactor for the demethylating process. So the TET1 gene, I think, is one of the, I know, working with some of my brain colleagues, they came to give a talk in our department that in fact methylation was dynamic, and as soon as they mentioned TET1, I looked it up quickly, and of course it's a dioxygenase, and it requires alpha ketoglutarate as a cofactor. So alpha ketoglutarate declines with age. So as people get older, their genes will get more and more methylated.
Right.
Yeah. So so that's I look at the big picture.
And interestingly, for example, in the aging and cancer, they are like cousins, because methylation is increasing, and then when you see in the old age more cancer like expectations. So it is not kind of random. I agree with you, like the big picture, because
well, that aging connection has come up with the announcement of the princess in England getting cancer at a young age, in her 40s. And so then there was a couple of articles just in the last couple of days saying that the reason for that younger people are getting cancer at a younger and younger age, and it's due to increased aging. So whatever that means, yeah, that they're actually accelerating the aging, which in my world is accelerating their methylation.
Maybe this, again, environmental factors, because then you start to see environmental factors heavily dense in these modern times. Maybe our aging is accelerated because of the environment.
Oh, and I completely agree. A very interesting group that are being studied are the people from Alaska, the native people from Alaska. So if you measure their methylation with respect to age, they are below the line. So if aging gives you a line upwards like this, the people living a more traditional lifestyle are below that line, and those who are living in the fast world of and not eating properly are in the elevated methylation range.
It's like very good, like right away visualization of this. Like, I agree.
Yeah, this is a really interesting area, and unfortunately, we can't solve that part of it, even though we know that modern life is not the best. Anyway, I'm sorry to have to end this conversation, but we're like way over time, and I think that just speaks to what an interesting topic and speaker it is that we want to.
By the way, before you leave, I just put the EPIC link. I forget to put on my slides, but you can go from this browser. I just put on the chat. You can visit EPIC on the actual website.
Yeah, and anybody watches this recording, it's https://appic.brown.edu. So APPIC, not EPIC, the more famous.
Exactly, it's not the But,
All right. Well, thank you, everybody, for sticking around to the
thank you
bitter
so much
end.
for your time.
And you can always
thank you.
contact Dr. Uzun if, afterwards, if you have any other questions. Have a great weekend. We hope to see you at the next Power Talk. Actually, next week's Power Talk will be the last of the spring semester. And then we don't have any scheduled until the fall. So, if you want to see the last one, come join us. And then we'll be sending out information about about in the fall. All right. Well, thank you very much. Have a great
Thank you so much,
weekend.
Bye-bye.
Bye.
Bye.